Okay, we're going to get started. Hi, everybody. My name is Eloise Barrett, and I'm the president of the Representatives Council. And I'm a second-year master's student in the psychological research program. I'm honored to serve as the president of GRC, which is a student-led organization dedicated to advocating for the needs and interests of graduate students here at CSUSM. This semester, or this year rather, I've been privileged with my Vice President Corey Jordan, who's sitting right there, to host a couple events on campus and off campus. We've hosted the Epic Wins fundraiser, Graduating Getting Hired, which was a LinkedIn fundraiser slash networking event. And of course, the Grad Slam competition, which I'm doing right now. We work closely with university administration, including the Office of Graduate Studies and Research, which is OGSR, to ensure graduate student voices are heard. And this year, it's been my great honor to be the president, and I'm really excited to be here today. We've got great events planned, so let's get started. And please allow me to welcome our Dean of Graduate Studies and Research, Dr. Charles De Leone, who will be sharing a few words today.
I don't know what.
Sure.
Just welcome to everybody. I am so happy you're here. Right now, I know many of you are thinking, okay, I got my premium. Let's stop for a minute. Just recognize the fact you're. Let's give yourselves a right I think the challenge for us today, as the academics we are, is that talking short is harder than talking long. If you put any professor up in front of a thing and you let him go, you can ask those who've done this before. It's very easy to babble on and on and on, as I am doing right now. What is hard is to get to the point in a very short amount of time and convey the information that you need in that manner. That is today's challenge. And I just want to thank one of you for being here today and actually bringing your stuff forward. I understand that I think the whole psychology department is here. At least that's right. Yeah. Or at least there's plenty of them. So I'm looking forward to it. And I'll add one more thing, is that for me as an individual, this is a treat. One of my favorite things to do is to learn. And so to get to hear what each one of you has engaged in and the research that you're doing, for me is an incredible treat. When I tell people my favorite thing about academia, it's being able to sit down and listen to a talk and learn something new. And so I am taking a moment right now, I may not get this at the end, to thank each one of you for what you're about to do. Because for me, this is a treat. So thank you. A little more things to take care of here, a couple more bits of business. I want to thank Lily for sponsoring this. I mean, this is a huge deal, and this is a number of years in a row that you've done that. Please, let's give them a hand. These events do not happen without judges, and so Dr. Carolyn Kaplan, Dr. Leonardo da Silva, Dr. Allison Badelsky, and Dr. Nathan Wright. Let's give them a hand for being our judges. But this one, the person who we truly have to thank today for this event and for being able to be here and have this happen for us, is sitting right there, and that's Nam Wayne. And please join me in thanking him. Put this together and organize it. She's the reason. And then last but not least, of course, where are my friends from GRC? Thank you. GRC actually has worked really hard, and they're the kind of ones who organize it and make it happen. So let's thank the GRC. All right. Without further ado, I think we're going to roll this over to you guys and think about those 3 minutes. So please turn it over to you guys. Okay.
Thank you, Dean De Leone. Hello, everybody. My name is Corey. I am the treasurer and vice president for the GRC here on campus. I'm also the winner of last year's graduate STEM category. It's we autographed that, but it's fine. But I'm here to give you a quick rundown of today's competition. We have 3 exciting categories today. We have our undergraduate competition, we have our graduate STEM competition, we have our graduate non-STEM competition filled with amazing student researchers. I'm very excited for it. Each presenter will be given 3 minutes to present on a research topic of their choice with a single slide to deliver said information. They will be given a 1 minute followed by a 30-minute warning, then conclude with time as they present. And today's scoring categories, being distributed by our excellent panel of judges, are in the areas of comprehension, content, engagement, and communication, because what is this about if not communicating research? So, and the important part, the reason we're here, there is a very generous prize today for the winners in each category, but more importantly, the winners from our graduate STEM and graduate non-STEM categories will compete at the CSU-wide competition hosted virtually by CSU Fullerton this semester. Our rivals at Fullerton. I don't know if we like Fullerton or not. But without further ado, we have amazing researchers to present. Can we make some noise for our first undergraduate? Technology, man. All right, representing the program of biotechnology, please make noise for Fahima Abdullah Saeed. Yeah.
So just wait for me to give you the begin sign, okay? Okay. Ready? Yeah. So every detective has that one cold case that always keeps them up at night. And for doctors, that case is triple negative breast cancer. Hi, I'm Fahima, and for the next few minutes, I want you guys to think of me as a private detective. And now here's the problem. Most cancers have locks on their surface that we can turn with a key or medicine to stop them. But this suspect is called triple negative because it lacks all the locks. And it is a master of disguise. It is highly heterogeneous, which is just a fancy way of saying that the tumor is not a single enemy. It is a mixed bag of different communities. And if we only catch the average suspect, the real mastermind can slip through our fingers and go on to spread the cancer throughout the body. And to solve this, I needed a way to watch the suspect without them knowing that they were being followed. And to do that, that's where we introduced the technology on the left side of the screen, which is the holographic microscope. So standard forensics usually requires us to stain or kill the cells in order to observe them. But we can't study a suspect's behavior if they aren't moving. So with holography, it allowed me to create a live 3D map like the picture shown in the middle of the screen to measure the optical volume, which is the mass and density of the cells while they're still alive, breathing, and trying to hide. But a detective is only as good as their data analysis skills. So I created a Python program to process thousands of these 3D images looking for 4 basic parameters, which are shown on the right-hand side. So I was looking for area, eccentricity, irregularity, and optical volume. And by feeding these 4 parameters into my computer, I discovered that these tumors aren't a random crowd. They're made of 4 different cell personalities. So first we have the uniform, and these are the ones that blend into the crowd and they appear standard. Then there is the irregular group. So these are jagged, active suspects that look like they're going to do something. And then I found the large and elongated cells that are stretched out, which is a sign that they are getting ready to migrate to other areas of the body. And lastly, there are the dense cells, which have the energy to divide. So why does this all matter? This matters because you can't stop an enemy that you don't know. And so by identifying these cell personalities, we're building a cellular fingerprint for each patient, and we're moving away from a one-size-fits-all medicine to more personalized. And doctors can choose the exact treatment that will benefit the patient. And that is the end of this case. Thanks.
All right, we'll now begin a brief Q&A from our judges. 2 questions, please. Please, Dr. Carolyn.
Hi. So you were talking about how this helps you look at live cells, correct?
Correct.
Yeah. And were you saying that this is just an instant that you capture, or is that like a time-lapse video for like a certain amount of time?
Yes. So we captured cell culture, so we captured them over a 48-hour time lapse, and then all the data that we got from the holographic microscope, I put it into my system to get the different light groups.
Okay, so when you get them, when you get this data, is it from like a snapshot taken, or are you observing their like movement and change over a long period?
It's from the 48-hour period.
Okay, so you'll see how they change over that time.
Yes.
Okay.
Have you found that any of these parameters, these 4 parameters you're looking at, correlate more or less with kind of how aggressive or invasive the cancer is, or is that part of what the
Good question. So right now, and we say that the research is in very beginning stages. I do want to develop this further, and I can see how it can translate into like drug development for other topics as well.
I think we do have time for one question from our audience if anyone is interested. Going once, going twice. Okay, thank you, Bahima. Let's give her a hand. All right, with that, our next undergraduate presenter of the day is Haley Birch from the biochemistry program. There's Haley. Make some noise for Haley, please. Yeah.
Can y'all hear me okay like this?
Yeah.
Yeah.
You know that use it for the recording.
Sure.
Good. The music, okay.
On.
Okay, I'm gonna do the. Okay, you got it. All right, a deal alter is a synthetically valuable mechanism that uses the diene and a dienophile to make new carbon-carbon bonds. This can often be the precursor for much more complex molecules, which provides a great application for the pharmaceutical industry. This reaction is often catalyzed with the Lewis acid, which forms a complex with the electron withdrawing group of the dienophile. Electron withdrawing groups, like oxygen here in the example at the top right, likes to donate its electrons, while the Lewis acid loves to accept its electrons, which form that complex. This pulls on the rest of the molecule, making it far more reactive to its substrate pair. This increased reactivity lowers the amount of time and energy necessary for the reaction to occur. However, Lewis acids are non-recyclable and often pose environmental harm. Metal organic frameworks are crystalline forest structures made of inorganic nodes and organic linkers. They have a high tunability to them, which allows for a variety of applications, including catalysis. As a fun little tidbit, moths actually just won the Nobel Prize in chemistry this last year. They also have inherent Lewis acidity to them thanks to missing linkers from the inorganic nodes. A fancier way of saying this is they have an unsaturated coordination of an active site, but I like to think of it as a metal cluster having open arms that are available to grab different molecules. This allows for similar coordination, similar Lewis acidic coordination as we saw for previous Lewis acids, while also being recyclable and environmentally benign. Moths have been shown to be reused anywhere from 3 to 5 times. And iron-based moths use iron clusters, which iron is very abundant and non-toxic. So my research focuses on addressing 3 things. First, to screen diene and dienophile pairs. Second, to screen reaction conditions. And third, to test different iron-based moths. I'm specifically interested in perfluorinated moths, which add fluorine molecules to the organic linkers, increasing the Lewis acidity within the framework. After screening 6 diene and dienophile pairs, the best reaction scheme that we found was according to the reaction scheme on the top right. Moving forward, we will use that diene and dienophile pair to then optimize reaction conditions. We will change the solvent for every reaction, but we will keep the temperature at room temperature and the reaction time for 1 hour. So far, the best reaction yield that we have used deuterated chloroform, which is CbCl3, and had an uncatalyzed yield of 35%. After catalyzing with an iron-based moth, based on row 3 of that table, we got a product yield of 53%. To make sure that the moth was doing the catalyzing, we then reacted the same diene and dienophile with iron trichloride hexahydrate, which is the inorganic building unit of an iron-based moth. This had a product yield of 10%, which shows us that the porous framework is doing the catalyzing and not its inorganic building unit.
Questions from our judges? You're not clear.
What would probably be your ultimate goal? Would it be 75% kill? 90% kill? What is it? What were you hoping to achieve?
That's a fantastic question. I think we always want to get the best yield possible, especially when we're looking at total synthesis reactions. We usually take the yield that we get from the previous reaction and move that forward. So we want to make sure that we're retaining the molecule as much as possible.
A couple questions, really neat project, and I'm the chemist, so. Were you able, or as part of this research, to also recover the mop and reuse it, and or what you've done in these experiments?
Yes, so we usually collect them off using filtration. And then after we've optimized conditions, we're also looking to see its recyclability.
What do you think with the iron fluoride that yield went down?
Yeah, very interesting. Obviously there's always error, human error that can play a part. We're hoping to retest these reactions so that we can get a more accurate yield that we're expecting.
Thank you.
Yeah, of course.
Okay, any audience questions?
Okay, let's do that for Haley. Thank you. I know the deeper words, he said.
Our next presenter today is JJ Ibarra from the undergraduate psychological science program. JJ.
If you find the rest of my hair, because I'm no redness. Hello everyone, my name is JJ Ibarra. I'm a research assistant in Spark Lab here on campus. We primarily work in implementation science, specifically for autism services. What's special about implementation science is that it recognizes the real gap between the research that we develop in labs and the communities who are actually trying to utilize that research. What that looks like is not only do we develop primary interventions that are meant to be used directly with people with autism, but we also developed secondary implementation interventions that are received by people like leaders. For example, principals at a school or a clinical supervisor in a mental health clinic. And what our intention here to do is to increase the effectiveness of these primary interventions. Now, this idea is something we refer to as implementation fidelity, which is measured through a few different means, including successful provider training in the autism intervention services that they're attempting to do, as well as looking at the actual clinical outcomes of the population they're trying to serve. And again, this is in an effort to close that gap. Now, what my project sought to do was to further emphasize community engagement by qualitatively coding interviews that were done with a group of leaders who received this secondary leadership intervention. And asked them the question if they thought there were any adaptations that could be made to this intervention that they received. And my research question was looking for themes or commonality in these interviews. And I have time to talk about one of these themes that we found, which was accessibility to training resources. And I think this is something that we can all relate to, the frustration that we get when we're trying to access essential information and we're blocked by something like needing to request permission from another person or being unfamiliar with the shared drive that might be hosting these materials. And this is a great example of the beauty of implementation science because it highlights things that might be missed, something as simple and practical as access to information when we're developing research interventions. Now, next steps to take on this project would be to implement these themes, all of them, into a brand new secondary leadership intervention program. And then again, measure implementation fidelity and look for continued improvements in provider training in our primary interventions and evidence-based practices, as well as, again, look at those outcomes in the community and see if they continue to match what we're expecting from lab work. This is important because implementing community feedback is a practice in cultural humility and recognizing that community members should be constantly involved in the process of developing research, as well as really getting the most value out of the work that's already being done, the systems that are in place in communities. And finally, just fostering a enthusiastic, supportive relationship between community members and researchers. Thank you.
JJ, any questions from our judges?
Give us a few examples of the interventions. I don't know if you've been present in anything, if there are any examples, if there's something that you've seen change during your work.
Yeah, so I was not involved in the development of these primary interventions or in the leadership training, the leadership intervention, which is the team's trial. However, I am familiar with what one of the interventions looked like, or one quality was there was regularly scheduled coaching calls between the leaders, for example, like a principal or a clinical supervisor, with researchers. So throughout the implementation of this secondary intervention, there was regular follow-up and check-ins where those community leaders would be able to ask questions directly to those researchers, again, with the intention of being able to fill those gaps.
How did you choose which institutes to work in? And like, how do you know you have a big enough number of those to adequately?
Yeah, so the sample that I had access to was 9 interviews. So again, I was not involved with these original studies. However, if I remember, I think it was mostly based on accessibility, but they tried to have a mixture of mental health settings, of school settings, and then diversity of leadership types within. But I can't speak too much more than that, unfortunately.
Okay, give it up for JJ. All right, coming up next to the stage, our next undergraduate presenter from the Psychological Science program, we have Caitlin Wagner. Caitlin, take it away.
Hi, my name is Caitlin Wagner from the Spark Lab, and today I'll be presenting on leader perspectives. When we think of preschool, we often think of learning how to share, maybe making our first friendships. It is in these best practice environments where we learn these developmentally critical milestones. We are often thinking of expulsion or exclusion, but for children with disabilities, this is their reality. One study found that the average age for expulsion is 3.3 years old. The same study also found that 1 in 6 children with autism will get expelled before they even reach elementary age. This raises a very critical question: if these practices are best basis for developmental milestones, why are we excluding some of the children who may need it the most? Well, the Spark Lab wanted to tackle this problem directly by looking at what ECEs are and what makes them inclusive. They found that, well, pardon me, ECEs are very important as they help form critical milestones. They help build peer relationships, school readiness, long-term success, social emotional learning, and they help get parents in contact with supports they may need. ECEs are often framed as universal, but inclusivity still remains inconsistent. Past research has emphasized a little bit more about caregiver perspectives. Little is known about leadership perspectives, and less looks at the holistic image of what truly is happening. In order to tackle this, the Spark Lab set a research project that was multi-stage. In this first stage, we are looking at leadership interviews. In order to do this, we interviewed 11 leaders in a semi-structured format. We then transcribed those and now are in the process of qualitatively and collaboratively coding them using Compiva. What we are finding are emerging themes that indicate that inclusivity or the lack thereof isn't necessarily an attitude problem, but more so a systematic barrier. We are finding that we are running into problems with money, financial resource, staffing resources. We're finding staffing resources, support services like behavioral therapists and speech pathologists, and also staffing training. Being aware of these systematic barriers is very critical as it can help form and shape different policies to help further inclusivity in these environments. Additionally, being aware and informed of these and shaping this policy can help make early childhood education more inclusive for children and parents who need it the most. Because ultimately, children, the last place where your child should be told they don't belong is in a preschool. Thank you.
Questions from our judges for Caitlin?
Really interesting topic, and you noted a few of the kind of reasons or shortcomings why some of these cases happen. Do you think it's mostly that students or caregivers or teachers don't, or teachers or administrators don't recognize an issue, or they do recognize the issue and just lack the resource to kind of help?
I think a lot of these leaders that we have been in contact with thus far have given many accounts that indicate that they know that it's a problem and they're aware of their inclusivity or lack thereof. I think all of them are aware, and a lot of them give accounts that they want to promote more inclusivity. They're just running into these barriers over and over again. It's just really hard to keep attention on that.
Just curious if in the interviews, you've heard anything about how these leaders are dealing with these barriers, if you find that those are effective strategies, or if you think that there was a different way of tackling them.
Yeah, absolutely. Some of these leaders have indicated that there are facilitators also for these barriers. They have given accounts that having behavioral therapists and OT therapists in their environment or early childhood programs has been very, very beneficial for both retention and keeping those children on track with their early learning success.
Okay, maybe one audience question. Okay, thank you again, Caitlin. All right, our next undergraduate presenter today, also a psychological science student. We really showed up today, huh? We have Mary Huerte. Mary, take it away.
Thank you, Corey.
Go, Mary.
Hello, everybody. I am a student from the Rockstar Lab, which is a child development lab that focuses on children's social cognition. So selective trust is a very important topic in developmental psychology because of its real-world implications. As you know, children tend to be incredibly susceptible to untrustworthy sources. They rely heavily on information from others, and it's important for them to critically think about who is a reliable source and who is not. Being able to verify an informant's testimony when they have the chance may greatly impact what information they choose to trust. And it's important for kids to think about what is a trustworthy source and whether they should independently verify those sources. So that being said, in this study, we play a sticker finding game in which children ages 3 to 4 years old have to pick which of 2 different cups has a sticker hidden inside. Before starting, we let them know that there are no tops on either cup so they can see inside the cups. And we ask them to look inside to find a hidden sticker. Then we introduce our puppet informant, Bear. We explain that Bear is tricky and will always hide the sticker in the opposite cup from what he tells the participant. Then we put up a divider between the cups and the participant so that Bear can hide the sticker. And when he's done, he tells the child which cup he put the sticker in. Then the participant has to decide which cup they think the sticker is in. So we record what cup they picked, as well as whether they decided to peek into the cups to verify what the sticker was before making their final decision. We do not prompt them to peek into the cups throughout the trials because we want to know if they will independently verify that information. So as for the results, so far, we see that they are picking the correct cup more often than chance, which is shown on this graph here. And so far, we have 21 recorded studies, with our mean age of participants being 50.67 months. Out of 84 trials, they picked the correct cup 63 times and decided to peek into the cups 42 times. This indicates that they are using their critical thinking skills in order to make an informed decision. And once this study is completed, we might gain some insights into what aspects of informants and information verification contributes to what kids decide are trustworthy sources. And that will be the end of my presentation.
Okay, questions from our judges?
Sorry, about the background of the children coming in, if you have that may affect their level of.
Yeah, so this study was really just very experimental. So we just wanted to see how it would go at first. We didn't have any specific hypotheses, but we did have our research questions up there. Yeah. So,
Are you thinking about doing kind of like a follow-up to this and seeing if those students still retain what they learned in the initial study?
yes, so we are actually planning on adding some more conditions to this study later on. This is a very early version of this study, and we're still kind of grappling with how to even run it. So these are the very early stages, but we are planning on furthering it.
Okay, audience questions?
Okay. Have you seen any variations based on variables such as like age or gender or something along those lines?
So far, not really. There's probably, if you look closer at the data, there might be a difference between like the young 3-year-olds versus like the older, 3 and 4-year-olds, but we haven't really looked into that yet.
Okay, give it up for Mary. Okay, is it a psychology student? No, it's not. Okay, we have Melissa Chico from the biological science program. Give it up for Melissa.
Well, in Lisbon, 3% how we usually impact plants and communities. So think about the last time you went to the grocery store, majority of this produce that you see are from a system called monoculture farming. This is basically where a single crop is being grown year after year. And although it's effective, majority of that it influences a lot of plant stress, unhealthy soil, and um, um, so the MIPA, so what if I told you that there was an alternative system? It's been here over 9,000 years. And let me introduce to you the MIPA. This is a mutualistic system. It is basically where a community of plants grow together, but we cannot forget about us. Without us, the MIPA wouldn't thrive. And so what we did was we cooperated with the botany garden and the food lab. And in this, we were able to cultivate a lot of the MIPA. And so what we measured was plant stress, carbon content, and microbial activity. With plant stress, we use a measurement called QM and physics table, and we use a tool called plant barometer. And as you see here is the understory for the MIPA, it was less than the monoculture. And for the soil, we measured with loss addition. This is a method where we're able to find the inorganic and organic carbon. As you see here, the MIPA was able to surpass the organic carbon. And for the microbial diversity, we measured with using ecoplates. This is a 96 well plate with 31 different carbon sources. And as you see, as a heat map, the yellow basically means that the microbes were able to digest the carbon source, and blue means that they weren't able to. So highlighted in red, the MIPA was able to digest the MIPA was able to metabolize a variety of carbon sources. And during the early breeding stages, that's when it was able to metabolize almost all 31 carbon sources. And as for the monoculture maize, it wasn't able to metabolize a variety amount. And so looking at these patterns, we were able to see looking through these patterns, they were aligning with the research that we see today. And for the research, we do want to see the microbes that are responsible for this diversity. Thank you.
Great job, Melissa. Questions from our judges?
Well, so, for this graph on the right, we're showing the 5 different categories. The variability seems really tight, which is good. How many individual measurements went into each group?
So we did 3 sugar bits for example. Yeah. Did that answer your question?
Yep.
Okay.
Okay, maybe an audience question?
So whenever I hear about field thoughts, it's always like maize, beans, and squash.
Yeah.
But in your picture, there's actually a lot more. And is that really what field thought looks like? It's greater than just those 3 sisters?
Yeah, so it's primarily known as the 3 sisters, but you can also incorporate a lot of different other crops. So like during maybe like in October or sometime I think later in this past season, we can start growing the shades. Like right now we did as well amaranth as well. So you can incorporate a lot of crops and they benefit each other.
Okay, and in that graph, the Milpa Mais, what was present? Was it more than just the three sisters?
Just kind of, what we did was in order to like, we have a lot of ground squirrels in the ethnobotany garden, so what we did around the perimeter was put a lot of, green onion starters, and that's kind of what helped it. But besides that, primarily we just had, actually, for the Mipa, surprisingly, we had a sunflower grow, which we're assuming maybe a bird dropped a sunflower in there. And so, yeah, so we did have a couple other plants that did benefit, and so I introduced a lot of pollinators to the garden.
Cool, thank you.
Okay, let's give it up for Melissa. Okay, next up, we have from the psychological science program, Marielle Dodds. Okay, Marielle. Sorry.
To start, I'd like you to think about the children's books you grew up reading. For many of us, this was our first impression about what the world was like. Who was going on daring adventures, fighting off dragons, and who was staying indoors, acting as the caregivers. Even though we didn't realize it at the time, these early narratives shaped how we developed our ideas of gender roles. This leads us to my current study, where we wanted to see how gender expectations are embedded in children's books. More specifically, how they can be found in the location and activity that the characters are doing. To do this, we coded a total of 24 children's books that were randomly selected from the Children's Books Online catalog. Of these 24 books, we used 4 of them to establish inter-rater reliability. What is that? Well, it's used to ensure that we were coding with the same types of standards and procedures. As for the remaining 20, we coded 4 pages per book, allowing us a total of 80 coded characters. Of these characters, we looked for 3 things: the gender, location, and activity. The gender distribution ended up being 45 male and 35 female. As for location, we looked at whether they were outdoors, such as in the wilderness, or indoors, such as in a home. Lastly, we looked at activity, whether they were doing stereotypically male activities, such as hunting or fishing, or stereotypically feminine activities, such as knitting and caretaking. This leads us to my results, where we found a significant relationship between gender and location, where 64% of males were portrayed outdoors, as 33% of females were portrayed indoors. We found a similar significant relationship with gender and activity, where male characters were 58% of the time presented doing masculine activities, while female characters were shown 69% of the time to do feminine activities. But why is this even important? Well, children are constantly absorbing messages about what is considered normal. With these repeated messages being embedded in all these storybooks, what types of messages are we sending to kids? Are they only allowed to be character A or character B? Our study hopes to promote that we push them to see more broader possibilities that go beyond just their gender identity. Thank you.
Okay, over to our judges.
I'm curious a little bit about, if you, focus on a specific language for the books you analyze, a specific country. I'm thinking of, maybe in different, literary traditions, you might have a space that is in one country dedicated to man, but in another one dedicated to another.
For the For the collection of our books, we utilized books that were easily accessible based on this catalog, and majority of the books ended up being classic books such as Jack and the Beanstalk and Cinderella, or books such as like folk tales, or like books like this one, where we could see it's a family, and it was more, I believe that was more of a Dutch book. So we did focus on primarily English written books, so we could code them. Yes.
Yeah, it kind of goes along with that. You might have touched on it already, but I was curious kind of what years these 24 books or the ones that you were published. And I wondered if there was a correlation to, kids' books from the 50s swaying one way and kids' books from the last 10 years, maybe a different way.
So for my book population, we utilized a range from
The oldest book being 1679, as it was an adaptation of Cinderella, to 2020, to one of our most recently published books. What we wanted to do with this study, as we were limited to just books that were free online, we ended up just utilizing those books. But what we hope to do with future studies is look into more presently published books and see if there's any improvement and progress in making more narratives that are more broader. So you mentioned that for 20 of the books, you just looked at 4 pages to each one. And generally, how did you decide on 4 pages per book? So for 4 pages per book, we decided on doing we used random.org. It was like a little random number generator to allow us to go make sure we were ensuring that there wasn't any bias in choosing the pages. And we decided 4 pages because as we had 20 total books and this is a project for a class, we wanted to make sure we were getting it on the deadline, allowing us to go have a total of 80 coded characters, which just in time made us in the deadline. Anything else?
All right, thank you, Marielle. Okay, our next speaker today is Miguel Kenyon. Both are no, but you have space. Thank you.
Hi, everyone. My name is Miguel Kenyon, and thank you for the opportunity to present my research today. So, what makes calcifying marine microalgae change life stages? Some of the organisms that help regulate Earth's climate are invisible to us. One of these organisms is the marine calcifying microalgae known as coccolithophores. Coccolithophores produce calcium carbonate shells by taking atmospheric carbon and locking it into their calcium carbonate shells called coccoliths. Upon dying, these cells sink to the bottom of the ocean and deposit in the seafloor, locking in that atmospheric carbon for millions of years to come. These calcium carbonate shells are also being studied by researchers for applications in drug delivery systems, optics, filtration, and many other medical nanotechnologies. Another fascinating aspect of coccolithophores is their unique haplodiploid life cycle, in which flagellated haploid cells, found typically in high light, nutrient-poor, high temperature environments, can reproduce asexually or fuse into heavily calcified diploid cells, typical of low light, low temperature, and nutrient-rich environments. The existence of these life cycle phases is well known by researchers. However, the triggers that initiate a transition between life cycle phases is much less known. To examine this, I studied nitrogen limitation, in which I plated these cells in seawater media replete with nitrogen, and also in seawater media deficient of nitrogen. During the first week, the cells all appear uniform in both conditions. However, after 12 weeks, in the middle panels, the cells in nitrogen replete media somewhat lose their ability to calcify, and in the haploids on bottom, form mucilaginous agglutination. After 12 weeks in nitrogen deficient media, much more significant morphological changes occur, in which diploid cells lose their ability to calcify, and cells of various sizes are visible in these conditions, which could indicate a transition in the life cycle. My future directions will be to focus on repeating this experiment and using a viability stain to ensure I'm looking at live cells, as well as to make sure they're indicative of a true transition in the life cycle phase. Understanding these conditions can help us to understand the effects of global climate change and mitigate them on our world's oceans. Thank you.
All right, questions from our judges?
Great images up here. And you have like these 6 that are your first one that's week 1 and then you 4 that's week 12. Are these just like shots from random samples or are like how do we know what sample they're from?
Yeah, so I meant to add labels on those, but So I did 3 of the triplicate wells of each sample, and these are just taken from one of the 3 wells of each condition. And the first week, both of the conditions appeared similar. They were calcifying the same, so there was still nitrogen in the media from carryover. But after 12 weeks, the middle panel is the nitrogen replete media, which is added nitrogen. And there's not much changes and not many different cell sizes. And then in the far left images, where there's total nitrogen starvation, is the main morphological changes that I wanted to note.
You mentioned the higher low nitrogen seawater, right? How do you know, or do you measure it, or how do you get that? How do you get high and low nitrogen seawater?
So we have a seawater media recipe that we use in the lab for most of the algae and the cultures that we raise them in. And I simply just made the media without adding supplemental nitrogen.
Oh, okay, you put this, you didn't know this was a sample from you.
Oh.
What's the time frame? Shorter or longer?
Of course. So it's kind of just thrown out because I was doing weekly observations, and I noticed that after about 12 weeks was when I started to see different cell sizes in the populations because they were isolated haploid and isolated diploid populations to begin with. So to see smaller cells in the diploid population could lead me to believe that there was a transition, or to see larger cells in the haploid population could lead me to believe there's a transition.
I was wondering if it's related to like a quarter of a year or seasons or something.
Oh, that would be interesting to try that. Thank you.
Okay, let's give it up again. Our next speaker today from the Mass Communication Program, we have Mila Duffy.
Hello.
Hi.
Oh, thank you. I might be able to zoom in. When I was 8, I watched a lot of Sex and the City. And although that now I know that that is not appropriate, it actually got me thinking when it came time to find a thesis for my communication theory class. So I decided to look into specifically specifically female friendship depictions in media, how they are potentially unhealthy and unrealistic that can cause women in real life now to feel even more lonelier. Whether it's Wings Club, Spice Girls, or those cool Monster High doll web show series that we liked growing up, they are all representations of the myth of the perfect cohort. Basically, a depiction of a friendship group that is free from the realism of real life emotional labor. Basically, we see perfect aesthetic women in their perfect lives, chic living, overall financially stable, and establishing their fields. It goes beyond them having their crap together. It's actually a systemic issue in media production as well. And we see this as a great, interesting way to basically disregard the real barriers of female connection that we see in a patriarchal society in relation to, say, the relational dialect theory, which says that relationships must be grown through tension. Basically, we skip the growing pains and we get right to already established relationships, which is not always a good learning opportunity for adolescents. The perfect cohort disregards basically the power dynamics that women go through and the barriers for connection. Think about things like social, economical, geopolitical, geography, time scarcity, or even being able-bodied or ability. And we basically do not share in media, TV, film, or music all about subjects like this. Scholars have already termed Scholars have already created the term the friendship recession when it comes to women. And I actually learned that that aligns with a actual recession, which is basically, there's already literature out there that when we're going through economic distress, we lean into traditional gender roles, which already have been proven to limit women's mobility and unmediated friendships and relationships.
In a capitalistic patriarchal society, we see it's perpetuated through a post-feminist individualism, which is a new type of relationship that women have with other women systemically, where they eventually just build their success on basically comparing themselves to other women, leading to that commodification of friendship that makes us spend more money the more lonely we are. It also perpetuates a patriarchal standard of what women go through that makes them basically perfect, free from fallibility, and also situated and happy being there. Thank you. Does anyone have any questions?
Questions from our judges?
I'm still formulating the question, so we're gonna do that together. You started your presentation mentioning Sex in the City, and something that I've noticed in and I think that you hit it in the nail when you mentioned the commercialization of these relationships. But looking at sitcoms and looking at how they seem to struggle to maintain the sitcom formula in this social media time and in this online connections that we made. So my question is, if you have had the opportunity of looking at any media depiction of these sisterhood happening online or happening not through characters that are interacting face to face, as in Sex and the City, Friends, whatever it is, but rather connecting through a mediated website.
Yeah, so that's actually a really interesting topic. When I basically talking about media depictions, I'm referring to fictionalized media. So that's going to be like TV shows, film, music, consumable media. What I would say you're referring to is actual real life connection. And with technology, there is a dabbling that we go for where basically we worry that like how connected is it versus how much it's harming us. So that's something you have to keep in mind, that some people have a bigger disposition to having social media be unhealthy for them. And for some people, rarely or another disposition for it being a greater form of connection, but it's on an individualistic perspective as well.
All right, thank you, Mila. Do we have any Sex and the City fans? Okay. 1, 2, 3, 4, couple. Okay. Maybe I should watch it. Maybe this is a sign. Okay, our next undergraduate participant today is from the biochemistry program. We have Oregon Sarah Belka. Take it away.
Okay, hi everyone, my name is Orkian Soropoka, and today I'll be talking about a small insect that caused a big problem in agriculture. So these insects are called cabbage bluebird, and they could cause up to $470 billion of crop loss every single year worldwide, actually. And many of us maybe not know that. During the larval stage, they eat a lot, and even though they don't eat, they're still growing. And because of how fast they grow, they affect a lot of crop loss. Yes. So just like human, these caterpillars have kidney, and this kidney called Malpighian tubules, and they have about 6-7 Malpighian tubules that work throughout the body. So these Malpighian tubules work by control water, salt, and remove toxin from insect's body. So Dr. Colosal lab here at CSUSM are looking whether changes microglobin in the Malpighian tubule changes in the Malpighian tubules. And to be part of this effort, my research in Dr. Ellie Beckett's lab, or also known as Blue Lab, are looking at the changes in Malpighian tubules and whether the changes are influence the comparability data. So to study this, Dr. Colson lab grow the caterpillars on food with and without antibiotics, and then they will carefully dissect out the gut, then pass it to my part. So I extract out the DNA and then use a library protocol called 16S RNA sequencing. And then I follow the early microbial protocols to do this method. And then I prepared this library, and because of this project is ongoing, so I don't have any data yet, but I hope to see the hexa that come out like this figure here that present different antibiotics and how these changes affect the comorbidities. So my goal of this project is to compare the data from my part with Dr. Colasola and then find out what response hexa and then could come up with further understanding with communities. So why does it matter? Well, because to understanding what's going on in the agricultures and it's involved with the integrated pest control management because if you want to control one species, you're gonna want to control other species and think about what affect all the living species as well. And I mentioned before that mouthpiece to build is responsible for remove toxin. So if antibiotics have effect with the system, they could potentially leading to avenue pest control. Thank you for your time and listening.
Okay, very cool. Any questions from our judges?
What part of the world are these, damage we put? Where are they?
Well, actually, my family is farmer, so I'm from like farmer family. So I'm from Thailand, and I've seen a lot of my crop loss that they affect, and also like they eat a lot of cabbage as well. So they are everywhere in the part of the world. So
And there, you said a big number, 400 something.
yes, 170 billion of crop loss.
Wow.
Okay, our audience?
Yeah, I've got one. Okay, we know that sometimes they'll use things like Bt toxin and stuff like that out on plants and stuff like that. So would some of these insects have adapted to their malfeasance tools can actually handle that kind of stuff and treat it? Or?
Yes, I believe so. That's why we're like specifically looking at the antibiotic, which is like cyclophosphamide and also sulfamethoxazole, and I think it's called amoxicillin. Then also we have control group. That's why we add control group disease. If the one that we don't feed, and other also don't feed with the diet, are there changes? And obviously I think it does because also Dr. Beckett specifically look at it and she like look at how the coastal change and the come from human, but there's a lot of antibiotics, so we want to figure out which is actually works.
Okay, thank you. Okay, guys, and with that, we are at our final undergraduate speaker of the evening. We have Sophie Thoreau. All right, so
Hi, my name is Sophie Theriault, and I am going to be discussing my research on enzymes, specifically for K26 biosynthesis and astrosporangium hypertensiveness. So K26 is an interesting drug molecule, specifically a natural product that is used to treat hypertension. So it's shown bioavailability or activity for this, but it's very hard to make in a lab. It takes many steps. The actual yield for it is not very good. So we want to look more into the biosynthetic mechanism of how the actual bacteria, astrosporangium hypertensinus, actually makes this molecule. So it's a specific type of natural product called a phosphonate. It has an interesting carbon phosphorus bond that is specifically bioactive in humans. And so it's present in a lot of drug-like molecules for natural products. Up above, there's some examples. Fosfomycin A and dihydrophos are both antibiotics that come from this carbon phosphorus bond that's interesting. So we looked at a couple different genes that encode for enzymes that we think are the actual enzymes that are making K26 in this bacteria, ON640, ON637, and ON638. So I produce these proteins and the enzymes actually, we tested them against assays for their activity. So 40 is the initial starting material of tyrosine into one of the intermediates AHAP, and then 37 and 38 seem to be adding this two other amino acids, another tyrosine and isoleucine. So our first assay that we ran was for tyrosine being turned into AHEP using ON640, which would be a novel mechanism for the type of enzyme that ON640 is. And we have the results in an EIC up here. So we have our control, which is the orange flat line in the front, the black line, and then the red line both have tyrosine present, ON640, and then PLP and ATP. So the control line does not have any tyrosine in it, and we did not see a peak or signal for this PLP tyrosine intermediate, meaning that ON640 would be working. So in the presence of tyrosine, we do actually see tyrosine and PLP interacting, which is promising for ON640 being the actual enzyme that is doing this reaction. Overall, this is important for making this drug more available for use in drug discovery studies or for future uses of other phosphates. Thank you for your time.
Okay, thank you, Sophie. Questions from our judges?
I think you say you produced these enzymes, the 1640.
Yeah, so these bioinformatically were selected, and then we got the DNA from a company that makes just exactly what DNA you want. And then in the lab, I actually did the protein expression in E. coli cells and then purified it to get our 3 different enzymes.
Tell us a little bit more about how you selected these to test.
Yeah, so a paper had actually come out about another phosphonate tripeptide, so 3 amino acids creating a molecule, and it had 40% homology. 2 of the enzymes had 40% similarity for homology to 2 of the enzymes that are present in the astrospharingeal hypertensionist gene code. So we selected these ones, and then 40 was very close and showed similarity to the PLP interacting enzymes, which is why we figured that 40 works with the tyrosine PLP carbon phosphate creation.
Okay, one question from our audience. Okay, that'll do it. Thank you, Sophie. Okay, with that speaker, we have concluded our undergraduate portion of Grad Slam. I just want to, can we please give it up for our undergraduate speaker? So, it's very exciting. And to start things off this afternoon, from computer science, we have our Chana Gopala. Take it away.
Imagine having to go through an open book examination on a subject which you have no clue about. So what would you do? To answer each and every question of the paper, you have to sift through an entire textbook to figure out where the answer is. So this is the state of the current AI architectures. So to answer a simple user question, the AI searches through the entire web to fetch your answer and then has to plan how to answer it to you. So what if it is possible to build an efficient AI architecture where you need not have to search through the entire web, but having shorthand code so that you can answer immediately to the user. Hi everyone, I am Archana Gaurava, a first-year graduate student in computer science. So today I'm going to present to you my thesis, which I am doing under the guidance of Professor Justin Morris. So as I mentioned before, to resolve this problem of AI, there is a promising solution called hyperdimensional computing. So it is a fancy word. This basically represents having shorthand codes of whatever is required for the classification task, or it actually prepares shorthand notes of whatever is required to answer your question. But there is a catch. Imagine there is a question on the importance of river banks. But you have gone through your book and the first thing which you have found is about financial banks. I mean the role of banks in an economy. So I guess the answer is going to be a disaster. You're going to write about River Bank is a kind of a bank, financial bank where you're going to withdraw your money, etc. etc. So even though we are having this hyperdimensional computing, it is very inefficient when there are similar words in different used in different contexts. So right now there are architectures. I mean in the modern right now there are modern architectures which actually captures this context, but they are very compute heavy. So the recent research in this field, they have come up with a solution. What if we are going to use the transformers, which is a modern AI, to fetch the context and pass it to this hyperdimensional computing so that it can do the easy task of classification. But if I mean using that paper, they have brought 25 times energy efficiency, etc., around 78% of accuracy improvement. But still, it is compute heavy, and you cannot run an entire efficient AI on your smartwatch or any low power devices. So this is where our proposal comes in. So we are planning to create a new architecture where the entire context or the relation between different words or classes can be represented by I mean in the regular way of how a hyperdimensional computing is done. So this is going to in this way we will be able to build very efficient AI systems. So some of the examples can be your smartwatch. If it is able to detect any changes in the different, changes in your heart signals and immediately, send the information to your caregivers or doctors saying that if you are going to have a critical condition. So such kind of things is not possible using the current technology. But if this particular architecture is realizable, it is going to make way to all such kinds of applications.
Okay, thank you, Arcana. Okay, thank you. Quick comment from our judges. Okay, thank you again. I think we're going to move on to our next. Okay, coming from the biological sciences program, we have Callie Savage.
So cabbage looper caterpillars are agricultural pests, and the order of lepidopterans, which includes moths and butterflies, are responsible for up to $470 billion in annual crop losses per year. They eat 5 times their body weight daily and grow 1,000-fold within their larval life cycle. And those larval life stages, because they're so detrimental, they need an efficient way to deal with all of the metabolic waste. So we have 2 kidneys, caterpillars have 6. So those mouth-feeding tubules are very cool to me. I love them. They can actually work in reverse, unlike ours. So if there's plenty to eat, they do option 1, where we call it reabsorption. They're going to source the ions they need to grow and live from their gut and bring it to their hemolymph. If they're in a period where they are moving to their next meal because they're tiny and it takes some time, or they are molting and they're not actively eating, they're going to be using option 2, which is secretion. So they're going to source their ions from their hemolymph or their blood, and then they can continuously be growing. So an assay I want to focus on today is the Ramsey assay. This is going to measure fluid secretion rate. So without their exoskeleton, that's what a caterpillar looks like, and that's what those 6 mouth-begin tubules look like. You do have to dissect all 6 of those out. They're not out free-flowing like the diptera. They're all tucked together and braided. So you have to dissect those out. We're going to then take all 6 of those into a dish of hydrated paraffin oil and a 60 milliliter droplet. We're going to tightly wrap one end of that mouth-begin tubule around a pin, treat it through some saline, and then that droplet you can see there is actually their fluid secretion or their urine. So we're going to do that
Every 15 minutes, we'll collect that droplet into a dish, and that's how we're going to get our data here. So this project specifically, which you're seeing, is voltage gated ion channel KCNQ, and is the voltage gated ion channel that I'm focusing on for my case today. The blocker we are using is linopyridine. That green line represents a control data set. The data that is plotted, you can see a significant reduction after linopyridine is added. And what that is telling us is there is a significant reduction in fluid after that blocker of the KCNQ1 voltage gated ion channel is activated. Thank you.
Okay, turn it over to our judges.
So this is very, very tiny. How do you think?
Practice, yes, semi-perfect. A lot of repetition, so it's actually very cool, if you ask me, maybe not everyone. We get to grow our own colonies of caterpillars. So you have to grow a lot of caterpillars, so you can practice a lot and get very good at it.
Do you use like tiny tweezers or needles
We use tiny forceps and iris tweezers, so my field likes to say the nanovexer rivals that of a neurosurgeon. So tiny instruments with tiny kidneys.
You're looking for magnification more than good.
Absolutely.
Question, really.
Yes.
What's your reproducibility like, if looking at within one? Or versus across different. You said you have 6 for each channel.
Yes.
Can you do all 6 and then do how many?
Yeah, good question. So the reproducibility, this is an N of 17, so that's say 2.5 caterpillars. You would never want to do one caterpillar because sometimes you get a caterpillar pre-block or you inhibit or activate a channel and it does something weird. So reproducibility, wanting to do it over and over with different caterpillars, is just going to make your data a lot more, not significant as an average, a lot more believable. I think. Yes, please. I've been doing it a little bit.
Okay, thank you, then Callie.
So we heard on categories. All right, our next graduate STEM presenter from the chemistry department, we have Christopher Lambert. All right, Christopher.
Hello, I'm Christopher Lambert. I'll be talking about the development of purine-based porous magnet. Porous magnets are typically made of a metal node and a molecular linker. In this case, I'll be talking about a transition metal as a node. And for the linker, I'm developing a purine linker. The reason for the transition metal is that in this specific porous magnet, I'm trying to create a paramagnetic field, which has a different application compared to magnetoid magnets that has a ferromagnetic field. And that's because it has an application for gas separation where you could think of it as like a bridge magnet as a paramagnetic the porous magnet. And you know how some metals can't stick to it, but other ones can. That's the same case in the porous magnet, is that the paramagnetic from the iron allows for it to attract like paramagnetic gases like oxygen, but not nitrogen, which is diamagnetic. So that's the purpose of a porous magnet over the other like magnetoid series magnets. And this is like a relatively newer field. So that's why I'm focusing on it. And I'm focusing on the purine because it's a rigid molecule that allows for higher stability. And I'm starting by making precursors for the purine base molecule. That's a target molecule, but the purine itself is just 4 benzenes. I'm making 2 precursors. The first precursor is activating it because it has, and then I have to functionalize the activated purine with an alcohol group, which is the middle group, which is the OH. I'm currently making and stocking up precursors because I need a lot to test a proposed method for it. And that's what I'm currently focusing on. And after I get enough of my material, I'll have to develop a porous magnet, and that's a proposed method to make a porous magnet based off of previous research using amines and an ion source. And that's just a focusing on what one part of it is.
Okay, Christopher, any questions from our judges?
That's a really neat structural framework that you're showing here for the forest. I think very cool synthesis. And it makes sense to kind of focus on iron as something that's not rare and it's cheap, you know, and makes it to be very accessible. Do you think, or is there a plan to try out other metals in the framework to see if you tune them a little bit?
Well, right now, we're just like focusing on seeing if it works.
Okay.
Once it works, we could think of like other transition metals with like paramagnetic capabilities, and we can compare like which one's stronger, which one's weaker.
Yeah.
But right now, we just need to see if it works first before we develop further with different metals.
Okay, one more question, maybe?
What I'm looking at, what is the long-term sort of application phase for this?
Well, right now we're like focusing on like gas separation. Like you could separate magnetic gases like oxygen. So that's like one purpose we're focusing on. So you could use it as a gas filtration, and that's like the one thing that we're looking at right now.
Okay, thank you, Christopher.
The next in our graduate STEM category, we have from the psychology program. No, from the biological sciences program, we have is Elizabeth Day White. Elizabeth.
Hello, I'm Elizabeth Demoya, and I'm a first-year graduate student in the Biological Sciences Department at California State University, San Marcos, and I'm a member of Ecomorph Laboratory. And for my thesis, I'm researching the San Clemente loggerhead shrike. A shrike is a carnivorous songbird, not a bird of prey. You may have seen them on nature documentaries. They're most famous for impaling their prey on sharp objects, such as thorns and fences, and storing them in larders to eat later. These particular shrikes are endemic to San Clemente Island. This small island is located about 65 miles offshore of San Diego County, and it happens to be a US Navy base. The island is special because it's home to more endemic species of plants and animals than any of the Channel Islands, and we still have so much more to learn about them. The San Clemente loggerhead shrike has been a federally endangered species since 1977 due to the island's history of habitat degradation that decimated the population of loggerhead shrikes, as well as the local ecology. Because of overgrazing and predation by non-native species, including sheep, goats, cats, black rats, and ravens. The good news is that there's a recovery program working to restore the species. I'm working with the San Diego Zoo Wildlife Alliance, and they operate a conservation bird breeding facility on the island that periodically releases young shrikes into the wild. And while the population has increased from a historic low, they still don't have a great system in place for tracking the health of the birds following release. And so that's where I come in. My role is to develop an automated weighing system incorporated into supplemental feeding stations already in use on the island. And the idea is that when the shrikes stop by to get some food, they will land on the scale, activating a trail camera that will capture an image of the weight reading. This data will be recorded and analyzed along with factors including age, diet, weather, season, and geographic locality. Body mass is an important metric for determining the health status of birds. With the system in place, researchers will have a way to weigh the shrikes without handling them. This reduces stress on the wild birds and keeps us better informed as to the conditions affecting their survival, helping to achieve the long-term goal of removing the shrikes from the endangered species list and eventually restoring the native ecology on San Clemente Island. Thank you for your attention.
All right, questions from our panel?
The feeding station show there, it looks like there's a little bit of debris that kind of falls into it. I think over time, things are falling up there and they're taking weights. Is that gonna sort of confound the weight measurements?
Yes, those are the dried mealworms. So the birds are supposed to be eating those, but should mealworms fall onto the scale, we'll be able to we'll have a time-lapse video so we can check the weight reading prior to the shrike landing on the scale and following, and then we can just do a simple subtraction.
Excellent.
I think I recall that you were saying that you were also trying to correlate more specific information about the bird, like their age and other information. How are you checking that if you're just getting like a video of a bird landing in the video?
The individual shrikes can be identified by their bird bands. And so with the records we have on the birds, we'll know their ages and we know their diet because they're eating, you know, we're the ones feeding them, and also we'll have we're keeping track of what time it is and the day, as well as there's an NALF station that records the temperature and the precipitation and all the various factors affecting weather.
All right, let's get up for Elizabeth.
All right, coming up next from the psychological science program, we have Harrison McCammon. Hey, Harrison.
Hello, everybody. My name is Harrison McCaney, and I'm currently a first-year student in the master's program in psychological research here at Cal State San Marcos. I work with Dr. Yasmin Sharifat in the Neurobiology of Addiction Lab, where we study a neurotransmitter receptor called the nicotinic acetylcholine receptor, or nicotinic receptor for short. And I'm here to present to you my project entitled Assessing the Role of Nacho in Cognitive Flexibility. Cognitive flexibility refers to an organism's ability to adapt to changes in their environment. It is not only evolutionarily important, but offers clinical significance, as this ability is disrupted in many neurological and psychiatric conditions. Cognitive flexibility is widely assessed using reversal learning paradigms, which offer a unique translational relevance, as variations of this paradigm can be used in rodent all the way to human models. In the version that I am using, I have mice that are presented with two levers, one of which will dispense a food pellet reward when pressed. The other does absolutely nothing. After a series of training and after learning has been established, the lever assignment is switched. The lever that previously dispensed a food reward now does nothing, and the lever that previously did nothing now dispenses a food reward. This allows us to see how well the mice adapt to this change as a measure of cognitive flexibility. Now, within our brains, much of cognition is mediated through these nicotinic receptors. And while there are a variety of nicotinic receptor subtypes, there are really two major players called the alpha-7 and the alpha-4 beta-2 receptor. Together, these two receptors comprise roughly 90% of our nicotinic receptors. And both of these receptors depend on a protein called nacho to form and work properly within the nervous system. Now, while prior research has shown that genetically removing either of these two receptors by themselves causes cognitive deficits in mice that relate to cognitive flexibility, no study has examined the effect of removing both of these receptors or removing the protein that both of these receptors rely on. So, in my current project, I have mice that were genetically engineered to lack the nacho protein, and thus the nicotinic receptors that depend on nacho to form, and I have normal mice. And we are assessing them in a cognitive flexibility paradigm using this reversal learning procedure. Our goal is to uncover whether nacho-dependent nicotinic receptors are necessary for mice to exhibit cognitive flexibility. This is an ongoing project and one that I'm very excited about. I really appreciate the opportunity to share this with all of you today. Thank you for your time.
Nicely done, Harrison. Judges?
Yes.
So you mentioned that when you're starting the process, you kind of have them learn about like the different levers.
Yes.
And then you switch them. What gauges that they've learned enough for then you to go to the next step and switch them?
That is a great question. So we have what's called stable responding criteria. And what is required is that the mouse must earn greater than 30 food pellets over 3 consecutive sessions before the levers are switched. And that can vary based on the mice. I'm currently running this experiment and I've seen it take as short as 5 to 6 sessions to as long as 9 to 10 sessions. And that's another component of this experiment. We're not only measuring how well they adapt to the switch, but how long it takes them to learn the initial rule.
That's gonna be my follow-up.
Does loss of nacho affect the production of those proteins?
Yes.
Or just the formation of the active?
So nacho is involved in these, so these receptors are formed of 5 protein subunits, and nacho is necessary for these 5 protein subunits to assemble together and move to the cell membrane to functionally express.
So they're still there, they just can't.
They have the material for the receptor, they're just not being assembled and not functionally expressing on the cell membrane. Exactly. Yes.
Are those nacho deficient mice otherwise normal?
Yes, that's a great question. So they do, that prior research has shown that they exhibit deficits in spatial learning and memory, which is related to cognitive flexibility. But the biggest concern is these mice have also demonstrated deficits, or they've also demonstrated hyperlocomotion, which could be something that plays a role into this experiment. But one way that we can assess whether or not this hyperlocomotion plays a role in this experiment is by also comparing their inactive lever presses. Because one explanation could be that they're simply pressing each lever more and more. And by forming a fraction of active to inactive lever presses, we can kind of isolate cognition as the mechanism rather than hyperlocomotion. Yes.
Recently, some neuroscience research has shown that you can use some really cool technology to stimulate individual neurons using light therapies and some assays like that. Is that on the pipeline at all for selective stimulation of this receptor?
I would love for that to be on the pipeline. I currently do not have the resources to do that, but I would absolutely love to get into optogenetics. Absolutely.
All right, let's give it up for Harrison.
Now I know they're all thinking, yes, Harrison was a middle school teacher. He thought that he got exactly middle school degree. All right, coming up next, also a psychological science student, we have Jake Stewart. All right, Jake.
Okay, hello, thank you all for being here. My name is Jake. Today we're going to ask and answer the question, do parasocial relationships appeal to lonely people? This is part of a project, a literature review I conducted during my first semester of grad school last semester. And the idea is that this will culminate in this project. So before we get too far into it, what is a parasocial relationship? It's a one-sided relationship that somebody forms with a medium persona. So a very relevant example would be like Swiftie. They're sort of characterized by this long-term investment and engagement in a relationship. There's no reciprocity, so they'll be one-sided. Contrary to popular belief, this lay belief, they're not adaptive at all. And in fact, when people engage in parasocial relationships, they feel good for it. They feel personal growth, they feel a sense of connection, they enter a community. So across the board, the idea that they lead to maladaptive outcomes such as forcing behaviors or stalking is just overwhelmingly not true. So given that, research has long wondered, well, how do lonely people view parasocial relationships? Do parasocial relationships appeal to them? Because with lonely people,
they have negative expectations about social interactions. And so what happens is they avoid them altogether. They expect judgment, they expect ridicule, awkwardness, so they abstain. Well, given that, it would make perfect sense, at least at face value, that a parasocial relationship appeals to them with its one-sided nature. Perhaps counterintuitively, we see actually that this is not the case. This could be because lonely people are treating parasocial relationships in the same way they're treating their face-to-face relationships. They have negative expectations, and so they're avoiding them altogether. However, there is recent evidence in the literature to suggest that maybe actually it depends. Maybe it depends whether loneliness will predict parasocial relationship engagement. What does it depend on? Well, there's evidence for identity. So people who feel lonely as a result of their identity, they then be more likely to seek out a parasocial relationship. For example, one recent study found that lesbian, gay, and bisexual adolescents were more likely than their heterosexual peers to seek out parasocial relationships when lonely. Perhaps more importantly, they were more likely to seek out parasocial relationships with influencers who share their identity. This is to suggest that they're consciously seeking out a parasocial relationship to fulfill a need that's lacking in their sort of immediate social realm. There's no option. So this is where my research is going to come into play. I've identified two groups in particular, those being sexual and gender minority college students, as well as underrepresented racial and ethnic minority college students. And the idea is that these groups, especially on a college campus, lack possible connections in their immediate physical social realm. And so what they may do is they may seek out parasocial relationships when feeling lonely due to their identity to then fill this need. So then, to answer the question, do parasocial relationships appeal to lonely people? Maybe actually, but it probably depends. Thank you.
Very cool, Jake. All right, judges.
How are you defining lonely people? Is it self-reported? How did you get to that category?
Yeah, that's a great question because, loneliness is something self-reported subjective experience, but that's actually where self-report is great in this instance, because it is subjective experience. So it's one's felt experience and it's their own perception of the quality of their relationships. So therefore, in this instance, a self-report is actually full of what? Anyway, that's the thing you get. So UCLA loneliness scale, robust scale, tried and true. Actually, choice.
Questions also on the self-selection part.
Got
it.
Quick question from the audience.
You mentioned how a parasocial relationship is not inherently pathologic.
Mhm.
That seems totally understandable. I'm curious that in your further research, are you interested in what qualities would cause a healthy parasocial relationship to develop? It's like pathologic stalking or obsession.
Yeah, that's very interesting. Potentially, yes. It's just there's so little evidence that they lead, to these, negative outcomes.
Okay.
What we find is just people who are very social in the day-to-day life, physically, happen to really like parasocial relationships as well. So not only are lonely people not engaging in parasocial relationships, but it's actually the people who are already fulfilled and their immediate social realm that are then seeking out parasocial relationships. Which can get so fascinating.
Thank you.
Thank you.
All right, one more time to go. All right, thank you, Jake. Coming up next in the graduate STEM category, we have from business admin, Jonathan Friedrich.
Hi, I'm Jonathan Burkage. I'm in the MBA program. And today there are 90,000 people on the wait list for kidney transplant. And about 10% of those people on the wait list die each year. And it's really sad in San Francisco, in certain areas, the average wait time is 18 years. So if you imagine having to spend all that time just waiting. And we chose this project and we chose to look at kidney transplant because it's a very unique organ. It's one of the ones that lasts the longest until it can be transplanted. So it affords us an opportunity to study some of the wait times. And we can also study certain things like travel and distance. So our approach, we're taking data and we are running a machine learning algorithm. We use logistic, linear regression, gradient boost, and ensemble methods to find out which attributes predict the long wait time. And hopefully what we're trying to look into is, can we recommend where a patient signs up for the organ donation? Thank you. And one of the things that's really exciting is because Oh, and one thing I forgot to tell you guys is that 24 hours can pass between an organ being received and then being successfully transferred. Heart transplant is only 4 hours. So 24 hours allows time for somebody from San Francisco to possibly travel to, let's say, Phoenix or other places within a reasonable time and have the success. So one of the things that excites me about this project is a very famous quote from an educator, Horace Mann, that said, "Refuse to die until you have accomplished something for humanity." And so this is why I'm inspired to be part of this research, and thank you everybody for your time today.
Thank you, Jonathan. Very important topic. Judges?
They're thinking on the 1 in 4 viable organs to start and
I forgot the paradox.
For some other reason.
Yeah, it's actually because of the inefficient process. And so that's another very key thing why we chose this is it's a shame, right? How do all these viable, and a lot of the time it's not viable, but the viable, that statistic blows our mind. And that's what we're trying to address, hopefully improve by the network. So people in San Francisco that are signing up, that something in Phoenix or Montana is being discarded, it's a flight or it's even a drive sometimes that if you expand that network, that donor network, you're able to assess those gaps and predict where, like somebody in San Francisco today, maybe sign up primarily in Phoenix. And then you're gonna have a better chance at getting an organ, and we reduce that the organs that go to waste. United States, correct, yes, thank you. Yeah, because that's another really interesting thing. The US has a lot of laws about paying for you could pay somebody. Other countries do that where family members or even there's a lot of ethical issues with that, but you could pay to procure a kidney. And so people might be in need and sign up for that. The US is not allowed for that.
Okay, one more time for Jonathan.
All right, our next participant today, coming from the Biological Sciences program, we have Julian Torres. All right, Julian.
Hello, everyone. Thank you for coming. Today I'll be talking about the Saltmarsh harvest mouse, which is an endangered rodent species that is completely restricted to the San Francisco estuary. So because the estuary is situated between the Pacific Ocean to the west and the Sacramento-San Joaquin River Delta to the east, there is frequent fluctuations in flooding levels and in vegetation structure throughout the estuary. Previous studies have found that these mice do not try to escape flooding events, which suggests that their swimming and climbing performance are important for their survival to some extent. Located in the northeastern part of the estuaries, this is Swin Marsh. This is where our study focuses on. It is made up of 2 parts. The large landmass in the middle is the Grizzly Island Wildlife Area, and it is separated from the mainland subsumed by a body of water called Montezuma Slough. Previous studies have found that even smaller bodies of water can be significant barriers to gene flow. This, along with the variation in vegetation structure, sets the stage for local adaptation to potentially occur within these mines. To investigate this, we measured swimming and climbing performance variables like velocity, stride frequency, stride length, in mice from Grizzly Island Wildlife Area, represented by this crescent unit. We also measured morphology variables like tail length, body mass, and we compared it to mice on the mainland, represented by Joyce Island, Hill Slough, Victoria Slough. This is a principal component analysis. It is the type of ordination analysis that reduces complex data sets into a few artificial axes that retain most of the variation or all the variation. The squares represent mean PC scores. The error bars represent standard error of the mean. And as you can see, there are significant multivariate differences between mice on Grizzly Island to those on the mainland. This shows that these mice are different in measurable ways. So the next part of our study is now we know that there are differences in these mice. Now we want to know why. So to get at this, we will be doing whole genome sequencing of these same mice. And with that data, we will link these differences in morphology and locomotor performance to their underlying genes. And then we will be measuring the strength and direction of natural selection, if there is any. This will give us better insights to how these mice are adapting to their changing environment. With that being said, I'd like to thank you all for your time and attention, and I'm happy to take any questions.
There is species that is close to the species account that is not endangered, that you can compare to multi-frame now.
That's a great question. So the question was, is there a species that is closely related that's not endangered? The answer is yes. In the same genus, there's the Western harvest mouse, which co-occurs with this saltmarsh harvest mouse. But we do have morphology and locomotor performance data for that. So we could use that. The problem is whole genome sequencing is very expensive. And in order to do like linking genes to these type of phenotypes, we need a large sample size, so we might not be able to get to that, depending on how much funding we get.
Any audience questions?
And, maybe just a comment about the multiple variables there. You said swimming, just which region had the best swimmers?
Great question. I'll answer that in two ways. So on the principal component 2 is negatively correlated with swim speed. So the faster swimmers, it's a lot lower on the graph. And on the map, Joyce Island and Hill Slough, based on our PCA, are the faster swimmers.
All right, thank you, Jillian.
All right, I checked and I promise our final psychological sciences presenter this afternoon will be Lizzie Lingsmeyer. Take it away, Lizzie.
Hello, everyone. My name is Lizzie Lingsmeyer, and I am going to present to you all my research on romantic conflict, sexual behavior, and mental health during the COVID-19 pandemic. So to provide you all with a little bit of background information, the spillover hypothesis is a theoretical framework that explains how external stressors affecting individuals can spill over into their romantic relationships and disrupt relationship functioning. We also know that relationship functioning is associated with the mental health of the individuals involved in a relationship, and this is of particular concern for the developmental period of emerging adulthood between ages 18 and 29, because mental health problems are prevalent during this time, and learning to navigate romantic relationship dynamics is an important developmental task for emerging adults. During the COVID-19 pandemic, there was an increase in mental health problems amongst emerging adults, specifically with regard to anxiety, depression symptoms, loneliness, and suicidality. In addition, during this time, romantic couples reported increases in levels of conflict and decreases in the frequency of sexual activity. However, we still do not know which pandemic-related factors influenced these relationship changes observed during this time, or how these relationship changes affected mental health during late stages of the pandemic. We hypothesized that COVID-19 risk perceptions would be positively associated with daily romantic conflict and negatively associated with daily sexual behavior 1 week later. We also hypothesized that daily romantic conflict and sexual behavior would be associated with anxiety, depressive symptoms, loneliness, and suicidality 1 month later. To test this, we conducted a path analysis using longitudinal data collected during late stages of the pandemic in the fall of 2021, around the time that coronavirus vaccines were made widely accessible. This diagram depicts our findings. You can see that risk perceptions were not related to the relationship variables of interest. And though sexual behavior was not associated with mental health, we did find a positive longitudinal association between daily romantic conflict and all 3 of these mental health outcomes. and the association between conflict and anxiety depressive symptoms held even when controlling for gender, cohabitation status, and baseline symptom levels. This suggests that conflict is a consistent predictor of emerging adults' anxiety depressive symptoms, which might exacerbate symptoms over time, independent of pre-existing symptom levels. And importantly, this research is important for informing interventions focused on conflict resolution, stress management, and dyadic coping strategies for emerging adults in romantic relationships, especially during times of elevated stress. Thank you.
Okay, questions from our judges?
Yes, that is correct. All of our participants were emerging adults between the ages of 18 and 29. Yeah.
Did you work with heterosexual couples and homosexual couples? Did you find any discrepancy in the numbers between one and the other?
We did not test differences based on sexual orientation. If I were to posit, I would say that I know there's evidence that individuals in non-heterosexual relationships experience higher levels of mental health problems than individuals in heterosexual relationships typically do. However, typically when we see differences in associations with relationship variables based on sexual orientation, these differences typically do not change the association such that it would become non-significant or change direction. So with that, I would say if there are differences based on sexual orientation, I would expect that they would be mean level differences, which may affect the strength of those associations, but I would still expect these associations to hold.
Hey, let's get up for Lizzie.
All right, coming up next, we have our second to last presenter in the graduate STEM category. We have Luke Etter. Let's make some noise for Luke.
Hi, everybody. Thank you to the last speaker for mentioning vaccines because that's the research that I focused on. Hi, I'm Luke. I work at La Jolla Institute for Immunology, and I study flaviviruses. Flaviviruses are a unique category of viruses because they're very closely related to each other, yet just different enough to elicit a specific immune response. So, flaviviruses are well-known viruses such as yellow fever, dengue, Zika virus, Japanese encephalitis, and they affect hundreds of millions of people every year. They do disproportionately affect developing countries and countries lacking proper mosquito vector controls. Now, this first map is a projected expansion map of mosquito populations that spread these viruses throughout the globe as of 2025. And you can see parts of where we are right now are highlighted in red, as seen by a dengue case in San Diego last year. Importantly, the mechanism of this disease affects people differently based on which virus they were exposed to previously. These viruses are very closely related to each other, like I said before, meaning that when our body experiences one of these infections for the first time, we develop an antibody response that can neutralize just that one virus. However, when you get a different infection by a closely related cousin of a different flavivirus, such as yellow fever or opposite dengue or Zika, your immune system thinks it's the same virus as before, and those antibodies that you made the first time no longer are able to properly neutralize that virus. This is shown very simply by those two viruses, one being coated completely with those black colored antibodies, and the other sort of being kind of not bound properly by those antibodies. Now, I ran a very simple experiment in my lab where I gave these mice either a cross-reactive antibody from a known patient who suffered from one of these infections, or just an inner antibody that's not reactive. And we saw a very significant increase in mortality in the mice that were given that antibody. Now, what that means for future designs, especially vaccine designs, that we need to tailor our specific focus towards immune targets that will not elicit these harmful effects. So very simply put, my lab focuses on developing vaccines which target the T cell response instead of antibodies. So thank you, and wish us luck in our journey in making this vaccine.
Okay, question for Luke?
So you're talking about how yellow fever, dengue, and Zika are very similar. In your study so far, are you like gauging, because you know that they're like very similar and that they can like have the same outcome, but have you noticed or are you planning to notice if there are any slight differences between the survival rate depending on which one you start out with?
It's a very complex process. Interestingly, we do have a vaccine that's very efficacious for yellow fever. Came out in the 1930s. It's 99.8% effective. It's fantastic. However, for dengue, we've known about this virus for hundreds of years, and we've been working specifically with complicated lab experiments for over 80 years, and we still don't have a good vaccine simply because it cross-reacts with these other viruses. Now, I guess to more specifically answer your question, we have sequenced these viruses. We do know their sequences. We know exactly which proteins are different, but it's actually the immune response that's more complicated. And the actual antibodies that you make, there's a very broad range of antibodies, and it's really about a niche balance of finding out which protein elicits an antibody response, and then how we can tailor our vaccines to give you those proteins without giving you cross-reactive proteins. So it's a really complicated process of testing each individual. It's really just trial and error. That's why it's taken over this 80 years.
Okay, thank you, Luke. I heard dengue infection in San Diego. My head popped up. I got a little scared. All right, our final presenter in the graduate STEM category from mathematics, we have Marvin Benya. All right, take it away, Marvin.
Hi, my name is Marvin. Today I'll be talking about some of the key concepts in my research, along with my goal and results. Now, to help me illustrate some of these concepts, first consider a day where you have multiple tasks due, just like this stick person right there. Now, these tasks could be anything simple like writing paper, cleaning your room, or responding to an email. And on these days, you may have more tasks incoming from either friends, family, coworkers, and so on. All these tasks or jobs add up to your workload. So in this example, let's say that responding to an email takes about 3 minutes, and all the other tasks take a longer amount of time. So in that case, if you're applying shortest time first, you would actually end up doing your email first. So of interest when you're considering all these tasks, shortest time first essentially makes you complete it makes you assess all the tasks and see which is the shortest time, which takes the least amount of time, and then has you finish that always first. So the great thing about doing that is the optimality property that shortest job first has. It ensures that we always get our jobs done as fast as possible, while also limiting the amount of tasks that we need to track. So this ends up freeing our mind and resources so that we can concentrate on other things. Now, my goal in my thesis is to find the equilibrium distribution of a system using shortest job first. So this distribution will help us characterize the behavior of the workload in the long run. In my thesis, we use an approximation model, and in the model, we have W sub t represent the workload of job sizes of X or less at time t. And that number line right there, we have the workload of job sizes X or less highlighted in orange. And of interest, we want to figure out how this relates to other workloads of different job sizes. in this case, in green, we have a workload with job size is greater, of Y. So in my thesis, as a result, we found a joint equilibrium distribution. So the joint equilibrium distribution of these two workloads and obtained an explicit formula for their correlation. Right there, we see that their correlation is equal to a coefficient C dependent on P, multiplied by that one minus the ratio of their sizes, X or Y, basically. So their correlation ends up just being a polynomial function.
Thank you, Marvin. Any questions for Marvin? Yeah.
Fascinating. This actually might be a way to really determine kind of mathematically, it's almost like a function of, I want to say diversity, in that people who come from maybe a less affluent background, right, social mobility, could be given an index, like the amount of kind of tasks that it's a really good way to measure someone's kind of how many tasks that they have to accomplish in one day, so many little things all at once. Whereas someone who maybe is a little bit, I'm not an expert in this, but more affluent or has less things to focus on, they may be able to, quote, accomplish more, when in reality it's just a variation in this kind of equilibrium. So it's I think potentially a fascinating way of measuring social mobility. That's not a question, it's just a really.
Right, we have a question, Dr. Wright.
Yeah.
Yeah, neat topic. Is there a capturing somehow of like relative importance of different tasks? Like the short one might be, less critical than the longer one. Is that incorporated?
So in this model, we're using what's in like the literature is referred to as shortest remaining processing time, shortest job burst. So in our model, we actually don't care about like their importance of like how it's going to affect our lives. We just care about assessing all the tasks first and realizing what task is going to take the least amount of time.
Okay, so all tasks are weighted equal, essentially.
Essentially, yeah. In the case where like two are equal in processing time, or they both take the same amount of time to finish, you just take one at a time.
Okay, thank you, Marvin.
Okay, and with that presentation, that concludes our graduate STEM portion of Grad Slam today. I believe we're gonna take another quick, for you.
Let's make our way back to our seats. I think we'll begin our graduate non-STEM category, our final category of the day. A quick reminder that the winner of this category will go along with the winner of graduate STEM to represent CSUSM at our CSU-wide Grad Slam competition at CSU Fullerton, hosted virtually. But yeah, let's get into it with our final participants of the day. Coming up first, representing sociological practice, we have Alexandra Ariza. All right, Alexandra.
So, hi, good afternoon, everyone. So my research is titled Silence and then Celosphere, Women's Experiences, Responses, and Digital Participation. And it focuses on incels and the ways that their ideologies shape harassment directed towards women in online spaces. And my research question asks, how do women experience and respond to harassment connected to incel ideology? And how does this shape their participation in online spaces? So to kind of explain briefly what an incel is, it stands for an involuntary celibate and it refers to a man who feels unable to form a romantic or sexual relationship with a woman, and therefore they often direct their frustrations and anger towards these women, and usually it's online, but it can also be offline. Much of the research on incels currently talks about their behaviors and ideologies, but less talks about how women have felt with the forms of harassment that they have faced online. So I hope that my research fills that gap. And to do this, I have been interviewing women about their experiences with the men that they describe to have incel-like attitudes and behaviors. So I'm approaching the term incel a little bit more broadly. And as you can see on the slide here, across my preliminary findings so far, participants have described encountering incel-like men who express misogynistic, racist, entitled, and aggressive or self-loathing attitudes. And these women have also described encountering forms of harassment online, such as sexualized insults, threats, stalking, or spamming, which then leads to emotional and psychological impacts such as fear, anxiety, or loss of safety. And in order to cope, women have begun to hide their identities online, reduce use of platforms, or just withdraw from certain online platforms altogether. Some participants also mentioned that since online anyone can be anonymous, so harassment is harsher in their end. And usually those people have fewer consequences. So platforms like Reddit or X that some of these women have used have become places where they feel more scared to share their opinion because of the experiences that they've had with incel-like men. So if women must constantly silence themselves just to avoid harassment, then who really gets to participate on online spaces? Therefore, in order to make these spaces more safer for women, the participants have suggested changing moderation systems and having a greater representation of women who develop moderation systems on these platforms or and who also design policy. So that's all I have. Thank you.
Okay, questions from our judges, Alexandra?
Yes. How do you find this event that we're getting? That's a really good question. So personally, I have put up flyers around campus, but mostly I used Reddit to put up a survey for people or women who are interested. But a lot came from like the snowballing off of like participants who may know someone who also has an encounter with themselves, but also some people that I personally know have had encounters with themselves. Yeah. Yes.
I was wondering if it's possible that social media interface with UX design, could that actually perpetuate hostile behavior online, or do you think it's moderately?
So just based on the interviews that I've already had, a lot of these women complain about like Twitter now, like X, after like Elon Musk kind of took it over, and they kind of feel like it's not as hospitable for women to be on there compared to it was in the past. So I think, yeah, the whoever is behind these companies or like making the UI, UX, I think that also contributes to like the hostility that I see with the bros.
Okay, thank you, Alexandra.
I was hoping I would go today without hearing Elon Musk, but anyways, our next participant in the graduate non-STEM category, we got Gafar from Public Health.
Hello, everyone. So my name is Gafar Farjat. I'm a grad student at the Master of Public Health, Education Promotion track. Before starting my study in the US, I lived in Germany, and in just 4 months here, I gained 15 pounds. Yeah. So I like to blame it on my cultural adaptation. It might have to do with things like unlimited soda refills, very large food portions. Yeah, but that led me to a very important questions. How do environment shape the way we eat and our health? And for children, school is one of the most important environment where children spend most of their day and consume large portion of their calories. And in the US, childhood obesity is actually one of the most pressing public health issues. Children who develop early obesity have higher risk for diabetes, for heart conditions, and many other chronic conditions. So what I found out, previous research focused actually on individual behavior. And so my research raised slightly different questions. What kind of food and wellness environments are schools creating for students every day? Some schools are actively, I would say, recommending healthy food, integrating nutrition education, have wellness leadership programs in their policies. But other schools, they have limitations in that regard, actually. And so the questions become, how do these differences show up in children health? And to explore this, I analyzed the best food forward data from southern Southeast Michigan across 8 schools, 194 students. And I am actually examining school environment factors as well as 3 early markers of obesity, body mass index, which reflects body weight relative to Asian sex, blood pressure as an early indicator of cardiovascular health, and HbA1c as a actually measurement of blood glucose regulation. So we, across schools, we observed noticeable differences in this health, I would say, indicators, and these patterns actually show how school context may influence health risk beyond the individual levels. So by expanding nutrition education, by strengthening wellness and actually leadership and limiting unhealthy food availability in schools, we can reach hundreds of students every single day. By implementing such policies, we can turn schools not just places where children learn math and science, but also for places where they can make lifelong health choices. Thank you very much.
All right, very nice, Gafar. Any questions from our judges?
I have, how do you separate out the influence of maybe the school from what's going on at home?
Yeah, that's one actually of the limitation of our analysis, and to be honest, that we do not have actually such, I would say, information regarding the beyond the school factors.
Are the schools from different socioeconomic regions?
Yes.
Do you see any correlations with like a general salary in that area or?
We have not reached that part of the analysis yet, but that's actually on our second phase.
We'll take a quick one from Christopher.
Looking at different countries, school nutrition to the American as well, or?
I actually did this in my initial literature review before starting this, because I moved from Germany, so I did initial analysis between like Germany and the US.
All right, thank you again, Gafar.
Graduates get the freshman 15. That's all. All right, coming up next for the history department, we have Joshua Hoover.
Good afternoon and thank you. The subject of my research project I will be presenting today is civil rights within San Diego. When some people think of the civil rights movement of the 50s and 60s, what often comes to their minds is imagery of the American South, as well as large metropolitan population centers like New York City, Detroit, and Los Angeles. However, racism, segregation, and discrimination in the workplace, education, and housing was all too familiar to America's finest city. In fact, what if I told you that San Diego was once labeled the Mississippi of the West due to its high concentration of white religious conservatives who fled the South due to their perception of supposed rising crime rates, declining property values, and urban decay? What if I also told you that San Diego was the hub of one of the nation's most active local civil rights chapters, known as the Congress of Racial Equality, or CORE? Or even still, what if I told you that in 1965, and again in 1969, our very own Mountain View Park, located in El Cajon, would be the site of bloody and brutal riots in which police responded with heavy force, leading to hundreds of arrests, dozens of injuries, and even in some cases, death. This is just a sampling of what occurred in the place we call home. Part of my ongoing research right now in my graduate studies is not just telling these stories, but also visualizing them from a bird's eye view. Utilizing important digital tools such as Timeline.js, Data Wrapper, and ArcGIS, It has been my intention to map out the history of both civil rights activists and their opposition within San Diego between the years of 1960 and 1970. I believe that by doing so, everyday listeners and viewers such as yourself would find that there is much more to our city's history than meets the eye. Additionally, it is equally important that any visuals, whether they be a map or a timeline, be crafted in a way that are appealing and eye-catching, that draw the viewer in and interest them rather than bore them, because let's be honest, to some of you, history is simply a series of boring events and dates. I hope that by sharing with you this localized research project of mine, it will give you pause and reflection about the importance of sharing these stories that occur within our local communities. As a historian, it is one's responsibility to tell all stories, not just some, especially when our preconceived notions of the past conflict with the reality on the ground. Thank you.
Very cool stuff, Joshua.
Just interested in what sources you're using. Are you going to archives in the city? What's the approach here?
Oh yeah, so there's one of the big ones, like this photo right here, is actually from the collection at SDSU. Harold K. Brown, who was actually one of the co-chapter leaders of the Congress of Core. We'll go with that. I forget it already. And so that's been good. San Diego Tribune also has a lot of digitized editions of the newspaper from the 1960s, where I'm looking at. So I also intern at the Escondido History Center. So that's something that right as we speak, I'm interested in looking in to see what they have too, since this is ongoing.
From our audience?
Given the availability of really easy to use creative tools like AI, generative AI, you see this potentially being carried into an online format of like potentially generative clips or some sort of like social media channel, which is able to portray these events visually.
So yeah, what I was talking about in this project, there are not so much AI, but some of the tools that digital history likes to use, like ArcGIS or ArcGIS, kind of maps out a visualization. I have one right now. It doesn't translate well to PDF. That's why it's not on the slide. But really where, you can scroll in and look at, specific markers on a map. It'll show you an expanded box of text. There are video clips that I do have, just of showing important events, altercations, demonstrations, stuff like that.
Okay, one more time for Joshua. All right, our next presenter today, on stretch here from Literature and Writing Studies, we have Maria Youngers. All right, Maria.
Hi, everyone. Happy to be here. In 2022, during my undergraduate studies, I founded a women-centered literary and arts journal called In Her Space Journal. For anyone new to literary journals, think of it as a digital space where women writers and artists can share their creative work. So anything from poetry, art, short stories, etc. From the very beginning, I had one clear goal: to create a publishing space open to all levels of writers and artists with no fees and no prior experience required. Fast forward to almost 4 years later, as the journal continues to grow, and transition into a nonprofit organization, I started noticing something interesting. Even though each piece was submitted independently, similar themes kept appearing across contributors. That observation led to my research question. What happens when I stop looking at creative works as individual pieces and instead examine them collectively to help guide the growth of the journal as it becomes a nonprofit organization? To make sense of all the data, I used a qualitative method called thematic analysis, which identifies patterns across texts. After analyzing a total of 107 submissions from 3 issues of the journal, 3 emergent themes appeared across the text. Memory and nostalgia appeared in 20 submissions. Shared identity appeared in 18 submissions. Community and relationships appeared in 16 submissions. So why do these findings matter? Instead of guessing what programs to create for the nonprofit, the creative work for contributors provides a data-informed blueprint for future programming. For example, Theme 1 suggests workshops centered around storytelling. Theme 2 points to programs like virtual showcases or open mic nights. And Theme 3 suggests formal mentorship programs that contributors feel supported and connected. The research shows that creative work in a literary journal isn't just art, it's insight. And when I examine the work collectively, it becomes a powerful way to build a sustainable community. Thank you.
Any questions from our judges?
Do you have enough data to see if the relative themes or how much they show up changes over time?
I don't actually. So these are just the top emergent themes, but I will say that there were 17 themes total out of the top 3, but these were just the most top 3. Yeah.
Can a journal, without the intention or a venue, influence what the writers discuss? I'm thinking of a poet, Ariana Brown, that has a poem that she says exactly how to win a slam poetry contest, and she jokes with it a little bit, like I can talk about all of these things, and I know that my audience will respond. So, kind of an open question.
That's a good question. Yeah, especially with something, for example, like poetry is kind of abstract to really code when using thematic analysis, especially. So I coded to my best ability, finding those descriptive codes, turning those codes into patterns, and then looking for broader themes. So that kind of like relates to even like the digital art submissions that people were submitting. I did the same thing with that, but looking at the visual imagery and evoking emotions from there and then finding those themes. Great question.
Okay, one more time for Maria, thank you. All right, with that, we are at our second to last speaker of the day. From the history department, we have Mary Handler. All right.
That makes sense in a second.
What is up, dude and student-ettes? I'm like so stoked to be here. My name is Mary, and I'm a history grad student, and before a grad student, I was an art media design major. So this is my project, Bombshell. Bombshell looks at the evolution of the blonde stereotype in the late 1980s and 1990s California beach television shows. The 3 that I'm focusing on are Baywatch, California Dreams, and Malibu, California, which are all fictitiously located on the coast of Malibu. Dumb blondes have captured our hearts for the last century, and we've laughed at Penny in Big Bang Theory, Kelly Bundy and Married with Children, Chrissy from Three's Company, and Jessica Simpson from the Chicken of the Sea tuna commercial. The Pretty and Dumb Blonde trope dates back to the 1925 book, Gentlemen Prefer Blondes by Anita Lewis. And the earliest, most impactful screen performance was by Jean Harlow in 1933 in the film Bombshell. Many scholars will argue that the blonde trope peaked with Marilyn Monroe in that era, but I disagree. I believe that the rise in the 1990s, bikini blonde, is actually the peak of the trope, and I have Pamela Anderson from season 3 of Baywatch as evidence. You don't have to be Elle Woods to observe the dominance of the blonde. I have turned my research into 5 quantitative categories to reveal deeper trends. The categories are blondes, bimbos, brain, bikini, and bombshell. Blonde is anyone with blonde hair that is on like the blonde hair spectrum. So that means dyed counts, dirty blonde counts, and bleached counts. Bimbo, is the flirty airhead, and brain is just like whatever the opposite of that is. Bimbo and brain are based on character behavior, while bikini and bombshell are based on costume. Bikini is anything that's a swimsuit. It can be a 1-piece or a 2-piece. But saying swimsuit is boring, so bikini is just much more fun, and girls just want to have fun. Bombshell is tight, figure flattering, skimpy, but not swimwear type outfits. To keep track, I individually tally every blonde visible in the frame, and that is from each shot, and that includes background actresses. I have discovered that episode content does play a huge factor in determining blonde count. This is consistent for Malibu, California, and California Dreams. California Dreams is on the screen for you. I analyzed the first 10 episodes of the first season, and the average runtime per episode is about 22 minutes. So when episodes take on stronger themes like racism or parental trauma, such as like divorce, neglect, or abandonment, the bikini count is lower. Themes like comedic dating or the liga has a crush have higher counts. And yeah, blondes appear dumber and skimpier to win over the hearts of men, while brains only show for comedic relief and rarely progress the plot. But Pamela Anderson has a net worth of 20 million, so is the ultimate blonde joke on us.
Right. Okay, any questions for Mary? JJ?
So you said that there was a peak of this trope, and in the absence of that peak now, would you say that has been replaced by another like aesthetic, or have we just stopped sexualizing women?
I don't think we've stopped sexualizing. I would say that, what I'm looking at is I'm saying the 90s is actually the peak of this trope, whereas a lot of people would say that Marilyn Monroe is just the blonde icon. But I think that the 90s actually created a new blonde icon, and that is like the bikini babe. And so it's the slow motion running in their swimsuits, just throwing their hair, and they're really ditzy, and they're just there for the guys to win over or flirt with. So, yeah.
Okay, thank you again, Mary.
I'm gonna start with the pretty, put down one, so I can send it back. That last one. Has our last participant True shown up? True, train? Okay, well, with that presentation, we are at the end of the graduate non-STEM category. Let's give it up again for all of our participants.